This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 353, recorded on September 4th, 2015. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today here in the TWiV studio is none other than Dickson Despommier.
Hello, Vincent.
Welcome, Dickson.
Thank you.
You know, we have a cloudy day today.
We do, and it's windy, too.
It's windy.
And how do we know it's windy?
Yeah, there's sailboats.
That's right. You were
tracking on the river.
Tracking a sailboat.
And it's hot. It's 27 Celsius.
But it doesn't feel as hot as it was yesterday, does it?
And we have 66% humidity, totally cloudy.
Yeah.
I mean, it's been sunny all week, hasn't it?
Yeah, we sort of need the rain. I wish it would rain, actually.
No rain in August, huh?
Almost none. A couple of thunder showers, and that's it.
Also joining us today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hey, Kathy.
You having nice weather?
Yeah, it's 77 degrees, blue sky with puffy clouds.
Lord.
And joining us from western Massachusetts, Alan Dove.
Good to be here.
How's your weather?
Pretty similar up here. It's 80 Fahrenheit, cloudy. Yeah. But it's supposed to clear up for the weekend, which I'm very happy about. So we're going to go camping.
Oh, good.
Where are you going?
Up to Athol. Don't laugh.
Athol.
There's nothing funny about Athol.
Sorry, I just had a tickle in my throat.
Actually, yes, I often get that when I'm going to Athol, especially when I pass the sign that says entering Athol.
Enjoy.
Yes.
Have a good time. You know, I have in my car, my dashboard temperature is in Celsius, right? Mm-hmm. And I just realized yesterday that it's screwed up to use Celsius because, and I know as a scientist I should use it, but the range is so tight.
Yes.
That's part of the problem. You know, it never really goes above 35, right? And really 0, -10 at the most. It's a really tight range, and I guess that's part of the problem. I know people have said that before, but sometimes things just hit you.
Huh.
Okay, but I will continue to use Celsius because I'm trying to get to know it.
I think my wife's has the option. If you switch it to Celsius, it gives you a decimal. So, but it's still a narrow range. So then you have to say it's 27.5 or something.
No, I don't get decimals. What kind of car is that? Is it Fit?
She just got a new Honda Fit, and I'm driving her old Honda Fit.
Well, I have a German car, and they don't use decimals. It will be 1 or 2 and nothing in between.
Nothing in between.
Well, I drive a Japanese car. What do they do?
Oh, that's what a Honda Fit is, right?
Yes.
You probably have decimals.
I have the option. I can go to...
But she has hers set to Fahrenheit. So
I can go to Fahrenheit, which I do.
This episode is sponsored by ASM GAP. And if you're wondering what GAP stands for, it stands for Graduate and Postdoctoral Opportunities, series of programs aimed at those individuals to hone their skills. This one is all about a webinar. It's from the Scientific Writing and Publishing Institute online. It is a webinar on scientific writing and publishing. This is going to be a webinar where you learn all about the scientific writing and publishing process. You're going to learn how to write a wonderful paper starting from the title and the abstract, how to make a good title. Like we do here at TWiV. You get Alan Dove. That's it. That's the answer.
Right.
Introduction, results, discussion, methods, figures, and figure legends, all about manuscript review. And you will leave this program knowing how to write a better scientific paper. And you may think, I've been publishing for years, I don't need this. But I went to submit a paper last week, and it changes like every month.
Also.
And you have to read all these guidelines, and frankly, I don't want to read them. So maybe you should just take this webinar. It's going to be run by Peggy Cotter, who's at the University of North Carolina in Chapel Hill.
And if you have a question during the webinar, you could say, Miss Cotter, Miss Cotter.
That's right. That's right. Welcome back.
Right. So you can see the syllabus for this webinar. The website is bit.ly/SWPI online. That's all one word, SWPI online 15. It's a 3-month, 6-part series from January to March of 2016. The application deadline is December 1st. So you have to apply and you have to be accepted. And then once you are accepted, you pay money as well. But it will be worth your money. It's a small amount of money to take care of all the expenses they have. So check it out, the Scientific Writing and Publishing webinar.
This looks like a lot better approach than when I was in graduate school and it just said write a paper. And
yeah, we had no approach at all.
Yeah, that's right. Well, we did publish, Alan, so you can't.
Yes, we did. We did manage to publish, but
it helps to write to begin with, I think.
And it depends on who your mentor is and
Sure.
how comfortable they are with the process. And
That's right.
Fortunately, you had a good one.
yes, I had a good one who knew a thing or two about
And,
publishing.
You and I published 2 papers, right?
I think we did 3.
Yeah, we had the commentary in science.
Right,
and
the
2
commentary in
data
science,
papers,
and we had 2 research.
Pretty good. Yeah, good papers.
Yeah, I had another one that was just about ready to go, but I just went ahead and put that in my thesis.
Speaking of your thesis, Alan, I wrote a web post this week about how you should listen to your mentor, right?
Yes.
And someone tweeted, my mentor asked me to build a 25-degree incubator, so I did. And it was just what you did.
Yes, I did that.
It wasn't you. It was actually a guy at UPenn. But he said I took a 37-degree incubator and I put it in the cold room, and
Yep.
that's what Alan
We
did.
arrived at the same solution.
It's very funny.
Oh, that's good.
Huh.
All right, we have follow-ups, numerous follow-ups. So let's start with Fiona, who writes, firstly, we would like to thank you for the segment on the current debate as to the mechanism of how viruses can become attenuated through whole-scale genome modification. We really appreciated the comments made during the discussion and enjoyed listening to the viewpoints of those on the panel. At one point during the discussion, it was suggested RNA structure could play a role in the mechanism of attenuation. This had occurred to us too, so we included a per-muted control in our studies, Atkinson et al. 2014, and eLife paper. Here, the peptide sequence was maintained, but we randomized the order of codon usage whilst preserving CpG-UPA dinucleotide frequencies. Permuting codon usage in this manner would destroy any undetected RNA structures within these regions of the viral sequence. The replication kinetics of this mutant virus were identical to wild type, supporting the notion that in this case RNA secondary structure is not involved in virus attenuation. A key point was made about the use of cell-free translation systems to analyze protein synthesis. In the absence of any cellular innate immunity-based translational control mechanisms, we and Burns et al. Carried out such experiments and did not observe any differences in protein synthesis between wild type and the different mutant viruses. Whilst the mechanism of
This is clearly a British writer.
yeah, whilst the mechanism, and the writing is all perfect, we can't criticize. Whilst the mechanism of attenuation remains unknown, data generated by Atkinson et al. 2014 demonstrated that high and low CpG-UPA mutated genomes were not sensitive to the cellular interferon response, but were, however, sensitive to the addition of the kinase inhibitor C16, thought to be PKR specific. The replication phenotype reversed following inhibitor treatment. With regards to the dengue paper under discussion, 1, the results in BHK cells, and 2, the effects of the JAK inhibitor 1 upon the attenuated phenotype of both the HMIN and the NS3 HMIN viruses, in accord with the C16 Atkinson data, suggest attenuation occurs through an unknown component of a cellular stress response pathway and does not support the hypothesis of attenuation arising from a defect in translation. Again, thank you for highlighting and discussing this topic. We think you all agreed it is certainly very interesting, and we hope your listeners enjoy the podcast as much as we did. And this is from the authors of the paper, Fiona Tulloch, Peter Simmons, David Evans, and Martin Ryan.
Cool.
Very cool. This was posted on the TWiV.tv episode. I agree that translation does not seem to be the issue here. And their results suggest perhaps that secondary structure is not. But then what is? Maybe this has to do with innate sensing, but there are ways to test that.
Well, the cell-free extract didn't show any.
No, there's no the amino acid sequence is the same. So why, would that be an issue?
Right. I mean, their theory is that it's this CpG-UPA ratio effect, but then the question is, what's the mechanism underlying that?
Yeah.
Right. Why does that have any effect? Yeah.
Now, I think this is quite interesting in light of the second email which has its own theory, and perhaps you could read that, Alan.
Sure. Mauricio writes, Dear TWiVologists, I can't explain my excitement while listening to TWiV 351. Just yesterday I met for dinner a wonderful physicist, Rudy Podgornik, who, in collaboration with his former postdoc, Luca Tubiana, have come up with a remarkable idea. In the world of physical virology, physicists and chemists studying viruses, there's a very hot topic which is of great interest to myself, how to characterize the physical properties of viral single-stranded RNA. This means finding metrics to characterize the overall size, the three-dimensional size, the degree of branching, the number of stem, loops, etc., of viral genomic RNAs and compare them with other kinds of RNAs. This quest has led to the observation by a few theoretical physicists and physical chemists that viral RNA molecules of icosahedral viruses are more compact than quote-unquote random RNAs with the exact number of nucleotides and base composition. These random RNAs are nothing more than viral RNA sequences that have been shuffled enough. Aaron Yaffe et al. Cites a paper, put this idea forward. Since then, both experimental and theoretical physical virologists have been trying to understand what this means. In the light of this quest, Luca Tubiana and Rudy Podgornik came up with a beautiful paper, see attached file, in which they took Aaron Yaffe's observation and asked the following question. How many synonymous mutations are needed to make a viral RNA look like a random RNA? In short, what they did was to mutate the genome of a huge number of viruses. All this work is theoretical. By following these rules. First, all mutations are only in open reading frames. They have to be synonymous. They can't be done in splice donor or acceptor bases, and they can't change the overall dinucleotide composition of the genome. With these restrictions, they found that mutating 5% of the genomic viral RNA is enough to completely disrupt the secondary structure of these RNAs, such that they now look like random RNAs. We and others have been trying to understand how the structure of the viral genomic RNA affects the packaging efficiency and selectivity of single-stranded RNA viruses. Hence, this observation turned out to be an exciting one. This paper, which I think is a beautiful one, opens the following question. Is there a third layer of information encoded underneath the genomic code? I know you might not be aware of the literature in packaging signals for single-stranded RNA viruses, but for sure there's a great debate on the nature of viral RNA packaging selectivity. It might be as well that for some viruses, packaging signals are not the way to go to ensure packaging selectivity, but it might be that the secondary and tertiary structure of the viral RNA dictates the specific interactions needed for such selective packaging. This is part of my work as a postdoc at NCI. The data of the papers you discussed in TWiV 351 go along with Luca Tubiana's idea. I don't want to get in the middle of the discussion between Simmons and Wimmer, but both experimental evidences are in agreement with Luca's observation. In other words, Luca Tubiana proposed that by changing the third base in the codons without changing the amino acids, they code for one but can completely disrupt the secondary and tertiary structure of the RNA. If by changing the dinucleotide frequencies you change the structure of the RNA and then the strength of the interactions between the viral RNA and its cognate proteins, then it's no surprise you end up with an attenuated virus. We're still missing the link between both papers, but it looks very promising. And Mauricio is a postdoc, I guess, at the NCI.
So the idea that secondary structure is disrupted is certainly not in agreement with Fiona's result, which she cited, right? But it might be contact between the RNA and proteins. And then if you look at these papers he sent, it's really interesting that it looks like a random RNA is much less compact than a viral RNA that may have evolved under conditions to maintain its compactness.
That's interesting.
So that could be the issue. Maybe by changing the codons, you change the compactness, and that attenuates the virus.
Right.
So inside the capsid, is there an association between the RNA and certain amino acid residues?
Yes, often there is.
Right. So the one that's got specific packaging requirements then would have a definable inner environment.
Well, in some viruses, as he noted, there is a specific packaging sequence which is rather short, which you can transfer to other RNAs and get them packaged. But for other viruses like polio, you can't identify such a thing. So it may be extensive. So that
it may be that the packaging signal is the tertiary structure of the RNA.
It may be.
Which I think is what he's getting at with an additional layer of information.
Right.
Or the overall compactness in some domains or overall. Yeah.
Yeah, if it gets too big, it doesn't package well, and then you get
Has anyone
attenuated
figured out how to
viruses.
watch viruses assemble in vivo?
What do you mean by watch? Do you want to watch them with your naked eye, Dickson? In which case, no.
Well, there are some interesting papers in Science this week suggesting that you might be able to do that someday.
Well, I don't know what you mean by watch. We can certainly watch assembly happen biochemically, right?
Yeah, but that's by extracting and watching at certain times. I want to see it.
That's why I asked you, what do you
Yeah,
mean?
with my naked eye. I want to use fluorescent labels and watch the thing come together.
Well, you can actually do that. There's high-resolution microscopy. You can do fluorescent labeling. Yeah, for sure. Dickson. No, not Dickson. Kathy. Kathy's
not Dickson. Not definitely.
Sorry,
I'm going to read Helen.
Yes.
Helen writes, Dear Vincent and co-hosts, I really enjoyed the discussion on TWiV 351 and wanted to chime in on one topic that came up in your conversation. We are most familiar with the flaviviruses that are transmitted by arthropod vectors to the vertebrate hosts, especially dengue, West Nile, and yellow fever viruses, which are transmitted by mosquitoes, as well as tick-borne flaviviruses. But there are also insect-specific flaviviruses that are transmitted vertically between insects with no vertebrate host. Furthermore, there are also vertebrate-specific flaviviruses that have been isolated from mammals but never from an arthropod. Many of these vertebrate-specific flaviviruses won't even replicate in insect cells in culture. While flavivirus host range is largely reflected in the phylogeny of these viruses, i.e. mosquito-borne viruses grouped together, tick-borne viruses grouped together, insect-specific viruses grouped together, there are several notable exceptions to this pattern. One interpretation is that there may have been instances in which a previously insect-specific flavivirus gained the ability to infect a vertebrate host, and additional instances in which a previously vectored flavivirus lost the requirement for an arthropod vector for its transmission. Understanding the factors that control flavivirus host tropism is critical for recognizing potential new emerging infections. Love the show. Keep up the great work. Best, Helen. And Helen is a faculty member at UNC Chapel Hill.
Who I interviewed a couple of weeks ago for a story.
Nice.
Where's that?
I don't know if
that going to be?
That's going to be in Nature Medicine. She might have discovered TWiV as a result of that. I'm not sure. We didn't talk about TWiV during the interview. But I guess these viruses come in many different flavies.
Yeah, flavies. That's right. Excellent.
Wrong.
Dickson, can you take David's, please?
I will attempt it. Dear TWiVniacs, regarding TWiV 351 and manipulation of dengue virus nucleotide sequences, there was a question as to whether DNA virus genomes had been studied similarly. I don't know about de-optimization, but there is an extensive literature on optimization of human papillomavirus gene codon usage. The objective is to study the less than optimal codon usage for humans of natural virus strains, presumably selected to attenuate replication and allow the virus to persist in its natural host, us. To me, that raises flags for anyone thinking about developing attenuated live HPV vaccines instead of the currently licensed recombinant particle. Friends, I'm not sure what else to call them, vaccines. It's sunny and an amazing 21 degrees this afternoon in Hershey. Fall must be on the way. Keep on twivin'. Dave. No, that's Dave is next. Yeah, Dave. No, two Daves.
Dave. That's Dave.
That's Dave. Okay, Dave. Way to go, Dave.
Yeah, there is. I found nice literature on this. The codon usage of the HPV genomes isn't always optimal for where you want to produce the proteins to make the vaccines. And so, for example, I found a paper where they talk about how codon usage is matched for keratinocytes, right? And that's not the same as a cell where you'd like to make, which is the host cell in humans, but not where you might want to make the vaccine. You certainly are not going to make a vaccine in keratinocytes. So they change it to match whatever the cell is. And that's a lot of work has been done to get better protein production in those cells.
But that's why you get warts.
That's right. So these are typically called virus-like particles, right?
Right.
Because there's no genome. They do look pretty much like the virus, but because they're free of the genome, that's why they're virus-like, and not recombinant particles. But you can call them what you want, Dave. Dave is a former professor at Penn State Hershey.
He's a professor emeritus, so he's technically
he's a former professor and now a professor emeritus.
Right.
Yes. A professor emeritus is like Dickson here. He doesn't get paid. He doesn't have And he writes.
Has no status whatsoever.
He was a professor, and then he came down with a case of
So
emeritus.
we had this discussion
Exactly.
years ago. He
we did.
yeah, but he's probably not going into work. He doesn't have a lab, that's for sure.
Well, Dickson comes into work.
I do.
So
he doesn't
I do.
really work. He just screws around.
I knew you'd say that.
He sleeps most
I
of
know.
the time.
Sometimes I take naps.
You know, it depends on the university. Like Dave, they might have given David an office at Penn State, which is great. That's what they do at a lot of places because you're considered a resource.
They don't do it here.
They don't care. You're not a resource.
Do
You're
not
just a
do it.
Because
liability.
you're not bringing in the grants.
Not bringing in the
Got it
grants.
No, emeritus, I think, just means that they don't throw you out of the building when you show up.
What does emeritus mean? Dixon, why did you ask for emeritus status? You have to ask for it and be nominated
you don't
and go
have
through
to
a
ask
process.
for it. You can be nominated for it, and then
Well, you had to
refuse it.
ask because no one would nominate you anyway. So,
Yeah,
what does it mean,
That's
Dixon?
a disparaging remark.
It
if
is
I
because
ever
you're
heard one.
disparate.
No,
Dixon, what does it mean to be emeritus?
It's recognition by the Board of Trustees that you were a worthwhile member of the faculty while you were functional.
What
and
the hell?
Therefore,
So you need
you're
that.
asking me a question. I didn't even finish the answer.
I mean,
and
that's
you're
an absurd-
jumping down my throat. Stop, all of you.
If that's really the explanation, it's absurd because of course you were functional when you were here.
no,
Why do you need
appreciate it. Appreciate it.
That's absurd. That's
It's
absurd.
not. It's not because sometimes people don't get it.
I think you have certain privileges as an emeritus professor.
Well, you're allowed to use the library, for instance. You can
Nowadays you don't even need
keep
right?
your what's a library. You can keep your email address
You can keep it anyway, can't you?
without having to pay. I think.
Seems to me that this emeritus designation
It's
is
an
a
honorary
little
recognition.
antediluvian, Dixon, if you know what that means.
I do. Yes, it's before Diluvia.
I'm not sure I would like to be an emeritus professor. I don't think there's
Well, I
any
hope you live
need.
long enough to be considered as for one at least.
Well,
Is there any downside to being an emeritus professor?
None, but there is a downside to being.
Can they draft you to come back to work?
No. There's a downside to being nominated and then not getting it.
Oh, yes.
yeah, then you feel badly.
Then you'll feel
Yeah,
terrible.
and your dopamine goes down.
Yeah,
and you get
mine was
depressed.
unanimously approved with 12 abstentions. There are 13 that serve on me.
So you can Google what does emeritus mean, and for instance, at the University of California, it's basically a synonym for retired and now, minus any teaching callback off
Right.
the university budget and onto the retirement plans. It's a combination
So
of age and years of service. This is because somebody wrote in and said, I've never understood the distinction except from the obvious fact that a professor emeritus is someone who is usually fairly elderly and has spent quite a bit of time in the university.
So you have to be awarded that by the Board of Trustees. This is not given. It's not automatic the moment you retire.
So the default is that you become a professor demeritus.
No. And in fact, associate professors that retire are not eligible for being considered to be emeritus.
You have to be a full professor.
Yeah, you do.
Well, maybe at your institution.
Yeah, I think it's different everywhere,
Well, that's true here.
which is just like tenure and what it means. You know, it's just silly. So the next one's from Neela. How would you say that? Neela?
Nela.
Nela.
I don't know.
Nella.
Dear TWiV gang, I have a very short question concerning the dengue paper that you discussed recently. If I understood correctly, Shennedow showed that when infected as young mice with the recoded viruses, they produced high levels of neutralizing antibodies. Since the changes are only apparent on nucleotide level, but not alter the protein once it's translated, the antigen should in theory be the same. So does this suggest that the attenuated virus gives the immune system more time to react and produce antibodies? Similarly, I'm confused about their conclusion that the attenuation of the viruses could be related to the synthesis of the polyprotein, its processing, misfolding, and or stability. How can the folding of the protein differ when the amino acids are the same? In addition, I was wondering if you could explain why mice are injected with the virus intracranially. After weeks of fluctuations between 15 and 40 Celsius, today we have 19 C and 65 humidity in Heidelberg, Germany. Since most groups here focus on HIV, and you could get the impression that it is the only important and interesting virus, I'm extremely glad that you are very considerate of other viruses as well. Thank you very much for everything.
We love them all.
What happens here is you infect mice with wild-type dengue, they die. And you infect mice with the attenuated viruses, the recoded viruses, and a good number of them survive, and therefore they are able to make antibodies because they're surviving. A dead animal will not make antibodies for long anyway. So that's the reason. And the intracerebral, I think that's just the way that you can infect mice with this
Right.
virus. That's the model that they have developed. Can't get it in any other way, and it doesn't replicate and cause any disease. So it's just a matter of practicality. Okay, we are back to Alan.
Okay, and I would just say I'm not sure about the misfolding of the protein. I would agree that if the amino acid
right.
sequence is the same, it should come out the
Yeah,
same.
we talked about that in the previous. Yeah, that's right. Yeah, we don't really think that translation is affected. Neither do the authors of the other paper.
So it might be a slowdown of replication.
Right, slowdown of replication gives the immune system more time,
that
and then
makes sense.
and then because the animal is able to survive,
survival
because
is
the
the key.
infection's a little slow, then survival is the key to the production of the antibodies.
Right.
Because otherwise, the mice just produce bodies.
Indeed.
Yeah.
Okay, so Anthony writes, thank you for reading my letter on TWiV. And Kathy has helpfully commented here that he wrote in about live animal markets in Minnesota and New York. I'll be amazed if any sort of surveillance is in place. My assumption is that health departments and animal control only act on complaints, and those reports will most likely be of superficial sanitation or cruelty issues. Union City, New Jersey was and might still be the most densely populated city in the U.S. Really?
Well, New Jersey is the most densely populated
It's
state.
the most densely populated state,
Yeah, but
I,
per what? Per square mile? Per block?
Yes, per square mile.
more densely
I thought
populated than New York
Manhattan
City?
was the
Yeah,
most densely populated per square
Which
mile.
one, Dickson?
Manhattan, the one we live in and work in.
State. New York is not a state. New York
Right, but
City.
he's saying
He
most
says
densely
Jersey
populated
City.
Well, I'm not talking about Jersey City. I'm talking
Anthony,
about...
let's
No,
get back to
Anthony's talking about Jersey City. I'm not.
He's talking about Union City.
Union
not-
City, New Jersey.
Let's back off of this a minute here.
Okay,
The
starting
most,
over.
here, list of states by population,
not-
rank, and land
Not
area.
state.
Yeah, but
Number
Anthony
one.
said city.
Number
Union
one.
City.
Yeah, but New Jersey is the most densely populated
state.
followed by Rhode Island, Massachusetts, Connecticut, Maryland, Delaware, and then New York.
Okay, but in terms of cities,
Well, you could
Right.
figure the most densely populated city might be in the most densely populated state.
No,
It's
no,
right.
no.
But I don't know about Union City. You go ahead and read, and I'm going to look this up.
Yeah,
Okay.
please do.
Union City, New Jersey was and still might be, and possibly we'll put brackets around the whole sentence. The most West. A significant demographic there travels to Central and South America, which I don't think we'll debate. Hoboken also is densely populated. The upper income individuals there often work in New York and can be expected to visit various destinations about the U.S. and around the globe. A sneeze here might mean that the world catches the flu. Back in the early 70s, during the outbreak of exotic Newcastle, my understanding was that commercial poultry facilities were all made biosecure. The recent Midwest spread of avian flu proved that if that was ever true, it no longer is. Containment did not happen. Is there any reason to expect a better outcome if or when the next disease infects people? Thank you for your time and energy. That goes into TWiV. And Vincent, you followed up here with somebody who should know.
Yeah, I asked Stacy Schultz-Cherry if surveillance of these live animal markets is done, and she said, yep, they certainly are. That's it. So they are. The USDA and APHIS keeps an eye on them, and Kathy's just racked
Kathy,
the
Sorry.
notes.
Kathy has dissolved the Union City debate, and it is
So,
appears
no, the most dense populated cities in the U.S.
Yes.
This is population density per square mile. Number
Yes.
one is Guttenberg, which is right next to Union City. Number two is Union City. Guttenberg is 58,000 per square mile. Union City, 52,000. West New York is 48,000. Hoboken is 39,000. Kaiser is 27,000. And New York City is 27,000 because it has a huge population but a lot of square miles.
Yeah, but New York
Yes.
City has 5 boroughs. So
Yes.
of the 5 boroughs, I'd say Brooklyn is atypical. Manhattan,
They have the whole thing lumped together.
I know they do. But
that's the thing, because New York City is one municipality.
yeah, but I think Manhattan holds the record for
If you
population
were
density.
able, if
You're-
you
you're
would
splitting
consider
hairs,
Manhattan a
Dickson.
single city, but
No,
Manhattan
but
is not
architects
a
talk
city of
about this
its own.
all the time.
Anyway,
But who's the mayor of Manhattan?
That's right.
Dixon.
No, there is a borough president.
There's a borough president, but the city as a unit
No,
is
I
all
got
of
it,
New
I
York
got it,
City,
I got
which
it, I
includes
got
all
it.
those
No,
areas out in the sticks
sure.
of Staten Island too.
Oh,
So
absolutely right.
that's interesting because, anyway, what Vincent found was a reference just for a listing for the United States
Yeah,
on
right.
Wikipedia, and what I found was a listing for the world.
world.
And they
And
did
the
supposed
world
to be.
listing is not comparable to what the U.S. listing is.
Well, Union City is down at 27 in the world, right?
Yes.
On your list.
Yeah, but it's the first U.S. one.
Yeah, it's
Yeah.
interesting. So Gothenburg, well, they're very close. Gothenburg and Union City are pretty close.
What is the leader in the world?
Manila.
Manila.
You can file that away in your folder. And the
I always
next,
preferred chocolate anyway.
And the next two of the top three are also in the Philippines, Pateros and Caloocan.
Really? That's interesting.
Wow.
Or Caloocan.
I always thought Mexico City and Tokyo were pretty high up on that.
They are the biggest cities in the world, but I don't think they're the most densely
Okay.
populated. I think because of the size of the land
Manila
area.
is 111,000 per square mile.
Yow.
That's twice as much as Union City.
Yow.
What famous actress made Union City famous in a movie? There you go.
Ooh.
Goodness.
Nobody knows, huh?
No.
I bet you our TWiV listeners.
Is it
know.
Marilyn Monroe?
This week
No.
in New Jersey, you're just tuning into.
That's right.
No, it's not Marilyn Monroe.
Not Marilyn Monroe. No.
Kathy, you don't know?
Oh, I'm not going to guess.
You going to tell us?
Telling me
should
yes.
I wait for people to email in?
Sure.
No, it's Union City with Debbie Harry.
Who?
Debbie Harry.
Debbie Harry? You don't know who Debbie Harry is?
Dixon, you know,
Debbie Harry?
do you know who she is?
I don't have a clue. What was her stage name?
Go away. It's a good thing you're not on a music podcast.
It is.
All right, who read that? Alan?
Yes.
So Kathy is next.
Uh-oh, I was just Googling Debbie Harry.
Christine. You don't know who she is either?
Christine writes, "Hello, everyone. My name is Christine, and I have been a weekly listener after being introduced to the show by a professor at Brigham Young University, Idaho. I am now a pathobiology PhD student at the University of Washington with an almost exclusive interest in virology. TWiV number 350, being shorter than usual, allowed me to go back to the beginning and listen to TWiV number 2 on poliovirus. In this episode, you referenced a paper entitled Virus Attenuation by Genome Scale Changes in Coding Pair Bias. Just as a quick refresher, the crux of this paper was using the idea of codon pair bias to synthesize the poliovirus capsid protein with over- and underrepresented synonymous codon pairs as a mean of viral attenuation for vaccine development. This paper was published in 2008, and I was curious if you are aware of any progression in using this method either for polio or other viral vaccines. Also, do you know if the idea of over- and underrepresentation of synonymous codon pairs has been found to be influential in anything other than viral attenuation, such as genetic diseases, perhaps? Thanks for doing this show each week. I love listening to your discussions and familiarizing myself with the weekly virology news." Christine wrote a few days later, "Dear TWiV, earlier this week I sent you an email concerning some questions I had about TWiV number 2 on polioviruses and the codon pair bias phenomenon. I had sent this email literally an hour before listening to this week's episode, which of course discussed this exact topic. Being episode number 351, I can't help but think, what are the odds? Anyways, sorry about that. Episode 351 was a great update and explanation. Thanks again for continuing this great podcast."
Here at TWiV, we respond very fast to listener
Yeah.
emails.
That's right.
Yeah.
That's exactly right.
All right, Union City is a 1980 American crime mystery. And Debbie Harry was well known at the time because she was in the rock group Blondie.
Blondie.
And if you don't know what their hits were, call me.
Not bad, Alan. Not
Excellent,
bad. Oh,
Alan. Excellent.
I know her by Blondie. I didn't know her other name. Sorry.
Yeah.
Okay, so I thought that was a cool set of letters there. And we have one more. I think it's your turn, Dickson, right?
It is. Barbara writes, "Dear TWiV and TWiVmers." Twimmers, that's right. Your podcasts on all the mind-blowing science going on today and are a weekly part of my mind. Thank you so much. Love you guys. Your recent TWiV episode with Michelle Banks made me wonder if you had ever heard of the Canadian conceptual poet Christian Boek and his xenotext experiment. A bit of background here, and then she gives a reference to which I haven't explored, but I'm sure that you have.
Why not?
Because I just read it. Love to hear what you'd think. Your devoted listener, Barbara from Philadelphia.
This is very interesting. So
This is.
the xenotext experiment with this fellow Christian, how did you say it, Dickson? Book?
Book.
He used the Canadian pronunciation.
It's an umlauted O, so it's got a little...
He manufactured a poem by making a DNA sequence, right? And then he took the DNA, he synthesized it, and he stuck it into a bacterium. And then the idea of this is he would then take the protein and decode it into another poem so that the cell would make its own poem. That's the idea. I don't know how exactly that the protein would be complementary. So that doesn't make any sense to me, because if you take the protein and make the amino acid, the codon sequence is going to be the same, or with degeneracy as what you put in.
Oh, I know what! I just hit me.
Yeah, tell me.
He's reading the antisense strand.
Okay, that could be it.
Right. So I was just looking at the sequence in this image of it.
But you don't need the cell to do that, right?
No, you don't. But he did it with the cell as part of the art project.
Right. So he spent 13 years doing this. Basically, it's just a random protein sequence, and it basically the bacterium he puts it in, it degrades it, which is not unexpected. And after 13 years, his funding ran out, but he's still, he said this is a Sisyphean effort, and I could see that this would be a cool artsy thing to do, but it didn't work in the end because you put any random protein sequence in, and it's not gonna do very well. There's no.
So it's an English
palindrome
encoded as amino acids.
Yeah.
But it's not a palindrome in English. It's a palindrome in DNA.
Right.
So you had genes will travel?
Right. So the poem, his statement to the cell is any style of life is prim, and the cell's response decodes as the fairy is rosy of glow. And you see the A in any is encoded by AGG. The antisense of that would be CCU, which is going to be read as UCC. That gives you the T for the.
Yeah.
So that's how he did it. It's a clever idea, and yes, that would be a heck of a lot of work.
I'm sorry it took 13 years, but
Yes.
you did get some funding for it. I bet you could get crowdsourcing today to do this.
Probably.
Anyway, that's pretty cool. Thank you. There are lots of examples of people using DNA to make various things. Like some people play, they assign a note to each of the bases and they play it, and that's interesting.
I've heard someone do that.
Some of them are pretty good, right?
Not bad.
All right, we have a snippet which was suggested by Andrea. Hi, TWiVitos. I like that very much. She found an article in The Guardian, which is a man found to have been shedding poliovirus, virulent strain of polio for 30 years. Someone else sent this by Twitter, and they called it shredding. Now, shredding is something else, right? Shredding is when you go down the snow really fast.
Right, at least they didn't change the vowel.
Exactly.
So Andrea,
because that's what he's actually been doing with this virulent poliovirus.
Have you seen this? What are your thoughts? 82F and still rainless in Seattle.
Gee, did you see this paper, Vincent?
Yeah, actually, full disclosure, I was the editor. I handled it for PLOS pathogens. So that means I got it, I sent it out for review, and I decided whether it should be published or not. And this is about Sabin vaccines, which when given to healthy individuals, they replicate in your intestines, you shed them, and then within a day or two they revert. They lose the mutations that cause them not to be paralytic, and they become paralytic. And these are called circulating vaccine-derived polioviruses, and they have caused outbreaks of polio in areas where immunization has dropped because they're fully virulent. And this is a problem. So one day, if we stop immunizing, these viruses, we got to wait for these viruses to stop circulating. And that's why the WHO has, in fact, said we got to switch to an activated polio vaccine for a number of years to make sure that these circulating vaccine-derived polioviruses go away.
It would have been good if somebody had told them that 18 years ago.
That's right.
Indeed.
What Alan is referring to. That he and I wrote a paper pointing out all these issues. But at the time, WHO wrote a response, and they said, this is not an issue.
Not going to be a concern.
Not going to be a concern.
Uh-huh.
But now it is, and they've changed to, they said we got to switch to IPV, so that's good. All right, now the issue here is that some people don't just shed for a month or two, but they shed for years. And these are typically immunodeficient individuals that don't make antibodies. They have a gamma globulinemia, okay? And you get polio vaccine in your first year of life, but you don't know you're a gamma globulинemic until after that. And so the vaccine virus replicates in these individuals. Sometimes it causes paralysis, sometimes it doesn't, but it just goes on and on and on. In this paper, they have an individual who has been excreting for 28 years.
I think you need to remind people what agammaglobulinic means.
So they don't make antibodies.
Yeah.
They lack the B cells that make the antibodies, and so they are susceptible to a number of many infections, and so they have to receive weekly infusions of pooled antibodies to protect them against various infections. And it's very interesting because these individuals, and I think there's 70 plus such individuals globally who have been identified since 1960. The virus is replicating in their intestine. There's no pathology in the intestine. Their intestine is fine. And if, occasionally the virus may get into their CNS and cause paralysis, that's a rare thing. In normal individuals, it's a 1 in 100 event. But this just shows that in the intestine, polio is well adapted not to cause disease. Anyway, this individual they followed, they could tell he had been excreting for 28 years because they sequenced the number of isolates. You can calculate the rate of change per year of the virus, and then we know how much poliovirus changes every year. We have a clock, and we can backdate it. And he'd been excreting since he was immunized. The viruses he excreted are virulent. If you put them in mice, it causes paralysis, and they're antigenically drifted. And so the point of this is that you have to keep immunizing, otherwise these excretors are going to infect other people. And whenever there's an area of the world where vaccine coverage goes down, and there have been many over the years, for example, there's just been an outbreak in the Ukraine of circulating vaccine-derived poliovirus, not from an immunodeficient person, but just from a healthy individual, because vaccination coverage had dropped to about 50%. Syria dropped into the 50s, outbreaks of vaccine-derived polio. So, if you stop using Sabin vaccine at some point in the future, which is happening, you still have these individuals, we don't know how many there are globally, that are shedding virus, so they're a threat. And furthermore, even if you identify them, there's nothing we can do right now. We can't cure them of the infection. So there are two things we have to do, which are suggested by the authors. First, we have to continue really good surveillance. You can pick up these shedders because the viruses have a signature that is distinct from other shedded polioviruses. So you could find them in sewage or in the environment. You could say, in this area there must be a person with this. Let's look a little harder. And
what do you mean by signature? I want you to explain that too.
So the sequence is different from circulating vaccine-derived viruses. For some reason, it evolves differently, and the antigenic structure of the virus is different.
Okay.
And secondly, we need to have antivirals to treat these individuals, right?
Right.
Because once you find them,
Sure.
you're not going to keep them at home so they can't infect others. You have to treat them. So right now, actually, a number of antivirals are being developed that could be used for these individuals. So that's why this is a very interesting paper. We've had other such individuals identified before. This is the longest one that we've identified. And it just underscores, as we get close to eradication, that these individuals are going to be a problem.
What interested me with this was the amount of news coverage this got.
It did, didn't it?
It generated, this thing was just, I saw the press release. I thought, okay, well, yeah, we knew this already. Good that somebody followed somebody long enough to prove that it is ongoing as we expected it to be. Nothing surprising. And then just my RSS feeds and Twitter and everything blew up with links to this BBC story, and everybody's covering it, and the man is secreting poliovirus. And I thought, okay.
I know, it's weird. So when I first got this, at PLOS, you have to decide whether you want to have it reviewed. And I thought, this, we've studied these individuals before.
Right.
But on the other hand, I thought this is a good time because it's not only the longest one, they've done a nice study, we're almost at eradication, right?
Right.
So it's good to underscore the fact that we have this as an issue. And I figured, the last of these papers came out before the echo chamber of the internet, and I thought this would be good. So,
yes,
anyway.
it really caught on.
Now,
just as a side note,
Yeah.
I happened to notice in the author summary they mentioned the projected cost of the polio eradication initiative at $16.5 billion, which is an interesting figure. It's that's almost exactly 100 F-35 fighter jets. You wanted something to compare.
Two
it
of
to.
which we saw fly by just a couple of days ago.
Oh, they were F-35s?
I think so.
Oh,
There's
okay.
split fins on the back of the tails. And
Okay, could have been F-15s, but
They were too small for that, I think. F-15s are bigger. Those are Navy jets, aren't they?
F-15s are National Guard, usually.
Oh, okay.
Now, in connection with this, H. Westster wrote in, he sent a link to a Washington Post article. It was entitled, Outbreak of a Rare Mutated Poliovirus in Ukraine Leaves 2 Children Paralyzed. And H. Westster wrote, has this happened before? Does the mutated poliovirus put the U.S. at risk? 92 degrees Fahrenheit, sunny, a triple H day near D.C. And I'll put a link to the actual ProMed Mail report. This is a circulating vaccine-derived polio outbreak. As I said, the Ukraine, the coverage went down, the immunization coverage went down, and that's why we have this outbreak. Because these viruses are shed by everyone who receives Sabin, so they're everywhere, and they're in all sewers.
Not rare.
So it's not rare. And I've actually tweeted the Washington Post author, and I said, this is not rare. Everyone sheds these, please. And again, this is not shed from an immunodeficient person. There's actually not yet any evidence. So this is the silver lining in the cloud of immunodeficient shedders. An immunodeficient shed vaccine-derived polio has never been shown to cause an outbreak.
Right.
So that may be good. Maybe they're not as transmissible as circulating vaccine-derived polio.
Right, because they do differ from the ones that succeed in circulating, so there's some kind of a leap that has to be made there.
That's right. Okay. We have papers suggested by Dirk. He writes, 2 papers on cyclic GAMP. I remember you followed this antiviral signaling molecule. They now show it can travel between cells using viruses. Thanks for the BPF. Best podcast forever. Cheers. That's why I picked these papers.
Also, I want to point out that I suggested them too.
You did?
Yes.
When did you suggest that?
Last week I
Anybody
did.
Nobody listens. Nobody.
But you didn't say it was the best podcast forever.
No, that would be self-serving.
When did you? I'm sorry, very sorry.
It's all right. Last week I said, maybe this could be at least a snippet, if not a paper, and then.
Oh, okay. Well.
Yeah.
Yeah. Okay. Thank you. I had forgotten.
It's okay.
Obviously, I have a faulty memory.
Yeah. So what should I do? I should write into TWiV.tv.
Right.
And
Kathy writes.
Camouflage my name.
Hey, wait a minute. Is that the same Kathy that's...
This is Dirk.
Okay.
No.
Best podcast forever.
Okay, I'll include that next.
Right.
Okay.
Now we talked about
first-time writer. Yes.
We talked about this system on TWiV 222. Jumpin' Jack Flash. It's a gas. Now, Dickson, do you know who wrote that song or who sang it? Or
it was the
performed
Stones. It was the Rolling Stones.
At least you know some musical history.
Stop it.
Why?
I'm not antediluvian. That's why.
Why? You were pretty
Come on.
ancient.
I have 2 sons. They both love music, and so I hear from them a lot.
Dixon, you know what dub is?
Dub?
Yeah.
Dub.
The musical style?
Yeah, it means that you record one voice and then you record again.
No, you're wrong.
That's overdubbing.
Okay, that's not what it is. It's a musical style. You can Google it.
I see.
Anyway, these are 2 papers published in Science. We won't have to go into both in detail, but I thought we had to do both at the same time because I think they're from 2 independent groups that came to the same conclusions. Yep. One is published in the same issue, right?
Published in the same issue, submitted on the same day, and both listing the same patent disclosure number.
They must have known about it.
So you think they were collaborating on the
well, but there's,
publishing?
as far as I haven't totally checked, but there's no authors in common.
No.
Yeah, there aren't.
Right. But I think when you go to meetings and you hear, and you go to Gordon conferences and you know who's doing work, and so they were probably informally collaborating for years, maybe.
Knowing about each other's stuff.
Yeah, exactly.
Right.
So
there was a paper in Science. There was a paper in Science. There was a lab in England. There was a lab in France.
There you go.
So the first one that I read, these are published in Science Express. These are these rapid publication things. And the first paper is Viruses Transfer the Antiviral Second Messenger Cyclic GAMP Between Cells.
And this is from the mostly British group.
That's right. And it's a little bit of Hanover, Germany.
Yes.
So the authors are Bridgman, Malfay, Daven, Partridge, Pangmeyer, Dong, Caver, Barrow, and Rehwinkel. So just to remind everyone, cyclic GAMP is a cyclic molecule with both G and A bases in it. It's synthesized by a protein called cyclic GA synthase. And cyclic GA synthase, cyclic GMP-AMP synthase is a sensor of cytosolic DNA. And on TWiV222, we talked about that. Yes.
We might accidentally slip and call it C-gas, the cyclic
Right.
G.
Yeah, C-gas sounds great.
Right.
C-gas sounds very good. So C-gas detects DNA. It then synthesizes cyclic GAMP. Then that binds to a protein called STING, S-T-I-N-G.
You know who Sting is, Vincent?
Yes.
I was actually looking at his Wikipedia entry just now because I wanted to make a title that somehow incorporated, what is it, Regatta de Blanc or Inagata de Viva? No, that's a different. Do you know that band, Dickson?
I don't.
Inagata de Viva?
I don't know that band.
That was by
But
Iron Butterfly.
I have met Sting.
Anyway, Sting. Yeah. And then Sting turns on interferon synthesis and establishes an antiviral state. Okay? Everybody okay with that little brief background? What does STING stand for again? Something inducer of interferon?
Right.
Stimulator of interferon
Excellent.
genes.
Stimulator of interferon genes. So in the first paper by Bridge...
Which are sort of the police.
Make a title, Alan.
Cell police.
Work on it. I did look up all the Sting songs, Dickson, but I couldn't come up with anything that would be a good title. You know? Maybe you can think of it, Dickson. Did you meet him?
Yeah, I had to sit down. It was nice.
One of the top grossing bands of all time.
Wow.
Indeed.
Yeah, they sold a lot of songs. Anyway, the first paper, they say, C-gas is important for sensing DNA, and HIV goes to great extents to hide its DNA from the cell so it can't be sensed. So they say we were trying to do experiments to understand the role of C-gas in HIV infection.
You just anthropomorphized viruses.
Yeah, I know. I always do that. I'm really sorry. I'm glad you were listening, actually.
I listen to everything you say.
And you're listening.
Everything you tell me. Every little...
Every move you
That's right. I will be watching you. I will be watching you.
Very good. Every sting you make. Yeah. That could be it, right? Anyway, so they make lentiviral vectors by... You can make these by inserting into cells plasmids that encode for the particles. And they use 293 T cells, which is a human cell line we've talked about before. And these cells turn out they don't produce C-gas, again, which is the enzyme that makes C-GAMP, right? So they make, what they have done is they modify the 293 cells so that they make C-gas or not. So now you have 293 cells with and without C-gas. So they make HIV particles with a VSV glycoprotein. Those are called pseudotyped. And they incorporate a green fluorescent protein gene in them so that they can track them. And they produce these particles in these 293 cells that either make or do not make C-gas. Okay so far? So we're making HIV pseudotype particles in 293 cells that either do or do not make C-gas. And then they take these virus stocks and they infect fresh 293 cells. They're fresh because... Well, you understand
They
this.
talk back to the...
They talk back.
To the culture.
293 cells don't make C-gas, but they do have STING. And they measure interferon induction. And they were surprised to find that the viruses made in the C-gas producing 293 cells actually activated interferon production in the 293 cells, but not the viruses that were made in 293 cells without C-gas. They did a number of experiments that we don't need to go into to address this, but it looked like the particles were transferring something to the cells that turned on interferon synthesis. All right, so remember, 293 cells normally don't have C-gas, they have STING, so the virus must be bringing something over to activate STING. So that's what's being activated here. They rule out that it's just a plasmid being brought over. They show that you don't need reverse transcription in the infected cell to have this effect. And so it looks like either the virus particle or something in the preparation. So these virus stocks, besides having virus particles in them, they could have little vesicles called exosomes. These are vesicles shed by cells that contain various components But they show that that's not the case. They can inhibit their production. And it looks like the activity that activates STIN goes along with the virus particles. All right? So they extracted small molecules from these particles, and they show that when you add those small molecules to permeabilized cells, you can activate interferon synthesis. And I want to just note here, they have this interesting use of transfection. You know, everyone uses transfection now to indicate the introduction of viral or DNA into cells. But now they're talking about transfecting cyclic GMP into cells, which I think is a stretch. I know it's a nucleotide. It's a cyclic dinucleotide. But what do you think of that? Is that transfection?
Certainly introduction.
Yeah. Anyway, they also they label cyclic GAMP with biotin. They put it in cells, which they then use to prepare HIV particles, and they show that it gets into the particles. They also use mass spec to show that the virus particles are containing cyclic GAMP. And here's the interesting number, Dickson.
Yeah.
The viruses contain 2.5 times 10 to the minus 17 moles per infectious unit.
How many people then calculated how many molecules per infectious unit?
Yeah. I don't know why didn't they tell us that?
Why didn't they just tell us that?
Yeah, what the hell is what'd you get, Kathy?
15 times 10 to the 6 molecules.
Yeah. Just multiply that by Avogadro, right?
Yep.
Yeah.
It just
I don't know why
cries out for it.
I don't know
Right.
why they didn't do it. Because, giving you a mole is not really...
Yeah.
To the minus 17. That's
But,
kind of,
if you learned it in 10th grade chemistry, you got to be able to use it for something.
Yeah.
Yes.
So this is all evidence that the HIV particles that they're making contain CGAMP. So they also show that a regular HIV, not a pseudotyped HIV, but a regular HIV produced in 293 cells will also incorporate C-gas into it. But now, here's the key, and this is also an issue with the second paper. In all these experiments, they're overproducing C-gas in the cell in which they're making the virus. Okay? And I talked with Steve Goff about this earlier. In the cells that HIV infects in vivo, CD4 cells, dendritic cells, monocytes, there is no induction of an interferon response because the genome is hidden. During reverse transcription, which happens in the cytoplasm, that happens within the capsid, and then that goes into the nucleus. So there's no activation. So it's not likely that in a natural infection that the HIV particles are going to actually package C-gas. So this is kind of a laboratory proof of concept. They go ahead and show that two other viruses, a murine cytomegalovirus and I guess that's in this paper. In the other paper, they use a different
VSV.
virus. They show that murine CMV will incorporate C-gas as well, so it's broader than just HIV. But I mean, it's an interesting concept, right? That particles can pick up a signaler of the innate immune response, right? But it's not. It probably doesn't happen with HIV in vivo in people, right? But these are all kind of proof of concept experiments. They also show that, let's see, they did an adenovirus experiment, Kathy, right?
Right.
Non-enveloped ad did not induce interferon, so they say that they're not picking up C-gas, right?
Right.
So maybe this is something that an envelope virus does only.
Right.
I was just wondering that if there's a signal for incorporation of the C-gas or if it's just randomly picked up. What did you say, 10 to the 6th molecules?
15 times 10 to the 6.
That's a lot of molecules, right?
15 million. Yeah.
That's a lot.
Now, that's per infectious unit. And Goff told me for HIV, an infectious unit is about 1,000 particles. So, it's still 10 to the 3 per particle. So it could, as these things bud out of the cell, maybe they're just capturing the cytoplasm and picking up some C-gas. Or maybe there's a signal that if there's a signal, the virus has evolved to have that signal, so there must be some use of it, but it's interesting. But it's unique, so this is a cool observation. The second paper is transmission of innate immune signaling by packaging of CGAMP in viral particles. It's by Gentili, Kowal, Katch, Sato, Lahai, Conrad, Boyron, Lombard, Duran, Kramer, Lowe, Delode, Terry, and Manel. They are in France at various places. And they are actually doing an experiment and got a surprise result. Now, I don't know. The first paper they said we were looking for this.
This
don't
just
know.
really this smacks to me of not a coincidence.
Yeah, probably not.
I mean, I'm not going to question who did what, but whenever I see something like this, and you do see this occasionally, two essentially identical papers come out from different groups at the same time, and you wonder, somebody heard something over beers and ran back to the lab and started duplicating it. And I certainly remember papers like this that I read in graduate school where it was obvious that somebody had copied somebody else, but you don't know who.
Yeah.
Well, just to clarify a little bit, I found the two approaches that came to the same answer to be really kind of different.
They are very
two
different
papers.
approaches, yes.
They're different. So in this paper, they make the statement early on that I kind of alluded to. In human dendritic cells, HIV normally evades sensing by C-gas, right? Because essentially the DNA that's produced by reverse transcription is shielded, so it can't be sensed. There isn't any particular evasion other than hiding the DNA. So they wanted to see if we fool around with C-gas, can we figure out, why it's so important. So they're using dendritic cells, which are derived from human monocytes. So you can get these from blood. You can differentiate them into dendritic cells. And they they were infecting these dendritic cells with various HIV vectors to study, sensing and so forth. Now, it turns out if you want to put a gene into dendritic cells using a lentivirus vector, you have to have this protein called VPX. It's a viral protein. If you don't have VPX, there's a cellular restriction protein called SAMHD1, which will inhibit the ability of HIV to insert its DNA into the genome. So they had done some experiments where they left VPX out as a control, and as they expected, they weren't able to get transduction. Transduction means the gene in your lentivirus gets into the cellular genome, right? But they found that the dendritic cells that they were infecting were still activated. In other words, they're making interferon, they're making cytokines as a way of preparing to interact with a T cell.
But they didn't get the gene.
They didn't get the gene. So they were really surprised. Why are these cells being activated? Without VPX, this shouldn't be happening. So that's what led them into pursuing this. And what's the signal in this that this virus is delivering that is activating these dendritic cells? And so from there, then they start to pick it apart. They fractionate the viral supernatants, and they basically show that the activity that leads to activation of these dendritic cells goes with viral particles. So virus particles can transmit the signals. If they make these viruses in cells with catalytically inactive C-gas, they don't transmit the signal. And basically, in the end, they also do an experiment here, which is a bit different. They instead of using VSV as the glycoprotein in these virus-like particles, they use influenza virus glycoproteins. It still works as well as that. And here's another interesting Englishism, if you will. They wrote this sentence: "We could not detect C-gas protein in the pelleted supernatants." Pelleted supernatants. That is a tough jargon, right?
Which was it?
So basically, they took the cells, supernatants, and they centrifuged them. Right. And then what pellets are the virus particles. And then the supernatant, they look, and there's no activity there, right? But it's kind of obscure. And it may be part of the compaction process for these very short, manuscripts that does that. Mm-hmm. So they do a variety of assays, very similar to the ones in the first paper, which show that it's the virus particle that contains cyclic GAMP. They do mass spectrometry, and they show that it's present there. They also want to know if, just like the first paper, if an infection with a normal HIV, not a pseudotyped HIV, would do this. So they do this in HeLa cells that produce C-gas, and they show that HIV particles, normal HIV particles, can pick up C-gas and transfer it to new cells. But again, these are HeLa cells which are producing C-gas. And as I said, in in a normal infection, the virus would not be inducing C-gas, so this probably doesn't happen. And they also showed this presence of C-GAMP in murine cytomegalovirus and a vaccinia virus used as a human vaccine, the modified Vaccinia Ankara strain. They found cyclic GAMP in those as well. So that's the extra virus that is not in the first paper. So again, the good data showing that cyclic GMP is incorporated into HIV particles, cytomegalovirus particles, vaccinia virus particles. But they say here towards the end, however, HIV-1 escapes cytosolic innate sensing by C-gas and thus is unlikely to contain cyclic GAMP upon natural infection. So this is probably more relevant to other viruses than it is to HIV. Mm-hmm. So I wonder, and they do too, what else is packaged?
Right.
That might be important.
Yeah, I was thinking, well, if ApoBec, what about Sting? Their experiments, testing Sting might not have worked.
Fit a few million molecules of this in there, then what else got put in?
Well, the cyclic GMP is pretty small.
It's a small guy. I guess it would be interesting to take highly purified virus particles of different viruses and just do mass spectrometry and see what's in them, right?
Yes.
So I talked with Ileana Cristea a couple of weeks ago, and I asked her this, is this a cool experiment? She said, yeah, it's a good experiment, but you're always going to have a lot of background. You have to make sure that it's not just stuff sticking to the outside of the particle, right? So you have to try and strip that off and see what's really inside. But
how conscientious do you have to be about stripping it, though? Because if it's stuck that firmly to the particle,
yeah, I know, that's the question.
It's going to get there.
Yep. So I suspect we'll be seeing more about this in the future. This is pretty important that you can put this very important signaling molecule inside of a virus particle, several different viruses.
Well, and these small signaling molecules have been understudied because they're hard to track.
Right.
So we follow the DNA, we follow the proteins, and that's all interesting, but there are lots of these little chemicals floating around in the cell that have important functions and that communicate things, and we don't know what they're up to.
So, Dickson, what do you think? Is it interesting?
I think it's not just interesting. I think you're absolutely right in your suspicion that perhaps there are other bioactive molecules. But I'm intrigued by knowing from the old literature that packaging of genomes in viral particles leaves little room for anything else to be there. So,
well, it depends on the virus. Some viruses, there's not a lot of room.
So which viruses would fall into that
category?
Kathy,
adeno, how much more can you put in an adenovirus?
Yeah, you can package 105% of the genome, and that's it.
Only 5%. Similar to polio, but for other viruses like VSV, the particle just gets bigger.
I see.
Right?
I see. Or pox viruses, maybe.
Yeah. So pox viruses have a lot of room.
Yeah.
And
they've got envelopes too, right? So
they do.
So stuff could be between the envelope and the virus
with
itself.
stamps.
Yes.
So that's what I'm thinking. I'm intrigued by the possibility. Okay. That's a paratenic kind of a passive transfer of
paratenic. What's that, Dickson?
Well, it's applied to houseflies and cockroaches and animals like that pick up stuff and then transfer them from one place to another without actually having anything biological going on in the vector itself. So here's nothing going on inside the virus itself, but here's a molecule that could affect the outcome of that viral infection in the next cell.
So to be paratenic, do you have to get inside the organism?
No, it could be on the surface.
So if I carry a virus on my shoe to Paris, am I a paratenic host?
You're a parasitenic host.
Yes. Flies
or a parasite.
On something and carry something on their feet to a new location or a classic example.
That's exactly right.
So I wanted to point out that both papers made the point that this could be important for vaccine development if you could load up viral vectors with cyclic GAMP. One put it at the end of their abstract, one put it at the end of their paper itself.
So the question is, did they know about each other's papers after they were accepted by Science, but before they got published so they could modify their discussions and more come into register with each
potentially.
Yeah.
But I'm trying to see if either one cites the other one, and I don't know that that's the case. I also had a point I wanted to make. Whenever I see the word exosome, I think, oh, wait, there's two things that are exosomes. And there's the exosomes that are vesicles that are secreted by mammalian cells. And then there's also the exosome complex, which is a complex involved in RNA degradation. Because I just saw, and it's unfortunate and confusing that there are two things that use those words. And then
that's a good point.
Yes.
The first paper, they did not find cGAMP in exosomes, but they did in the second, right?
well, in the second paper, they say in extracellular vesicles.
Okay.
But they use different cell types too, right?
I think the
HeLa
second one
versus-
used monocytes.
Yeah, DCs.
Transformed monocytes to dendritic cells.
Yeah.
So, but that part did confuse me a little. Is it in exosomes or not? And then I thought, well, they call them extracellular vesicles, so maybe that's why.
It's an important point, because if they're in exosomes, then they're just being picked up by the budding process, right?
Mm-hmm.
So whether it's a virus or an exosome shouldn't matter. So that would say the virus isn't have any specific signals to pick up the cGAMP.
Right.
Okay, we done with these? Anything else? All right. Alan, I think you should take that first email.
Sure.
Because you like
yes.
to read.
Well, he annotated it.
I annotated it a couple of weeks
okay.
ago when it first came up in the queue. So Wildheart writes, "Hey there, folks. I've copied and pasted some comments that were left on a Raw Story article about vaccination, and some of these arguments are not ones I had heard before. So I thought I would send them to you folks and see if you have any comments I can use to back these folks to use back to these folks. The truth is that I'm not a scientific person, and so I can't argue a lot of this back. I just know that I believe vaccines are safe, but they do name someone who seems to be a good, knowledgeable person, question mark in parentheses, so I hope you can deal with this. I also wanted to make another quick comment. Whoever it was that stopped you from using FU for follow-up needs to move on, and you guys should bring that. I get tired of these holier-than-thou types ruining just about everything. There is no need to over-censor your comments and stuff. Thanks in advance.
Thank you for reading that.
For your help refuting this.
This blankety blank blank.
Okay, so Wildheart has pasted in this comment from, I guess, I don't know what Raw Story is, but I assume it's a news site. And I would just point out that if you have spent much time reading the comments on science stories online, stop. Just stop reading the comments on science stories online. It's completely degenerated into stupidity. There is not enough of value in those to waste your time on it. And this thing that Wildheart came across is exactly an example of the type of nonsense that goes. Oh, I'm sorry, Gary is Wildheart's. Why is this person named as Wildheart at the start?
I don't know.
Okay, well, Gary wrote this. So this screed that was apparently posted as a comment is a bunch of standard anti-vaccination stuff that starts off by talking about how cultured cells are sometimes made, and then extrapolates into how this raises the question of how these vaccines that are made in cultured cells could have weird effects on the human body, which is just stupid.
By the way, he says that most vaccines are used in cancer cell lines, which is just wrong.
Right, which is wrong. I mean, it takes bits and pieces, and it links to an FDA document, which is on an unrelated project, and then takes little bits and pieces of sort of truthiness and pieces it together into, oh, gee, I'm just raising the question of could these give you cancer, which is not what they're doing. They're trying to plant an argument in your mind that, oh, gosh, vaccines cause cancer, which they don't.
That's right.
So that's a very standard extrapolation to absurdity. They subsequently slip in a reference to fetal tissue for a little added dash of stigma there. And then they bring up the flu narcolepsy story that we talked about extensively, and claim that that's part of a whole
boatload
of scientific evidence that vaccines induce autoimmune diseases, which is a straw man argument and a load of BS. There's no boatload of scientific evidence that vaccines induce autoimmune diseases.
Can I add one thing?
Go ahead.
So he says, we make these vaccines in human cell lines. Who knows how they're going to hybridize with our own cells? Well,
right.
There's no cells in the vaccine.
There are no cells in the vaccine, and it wouldn't work if there were. And then cites a textbook.
Are they gluten-free?
Yes.
GMO.
Yes. Cites a textbook called Vaccines and Autoimmunity, which I don't know this book. It sounds like a credible book. Talks a little bit about the book and the author, and then mentions a conference. I Googled this conference, and I'm pretty sure they got the name of the conference wrong. But then they describe and are claiming something that is supposedly said at this conference, but we don't know because, of course, there's no transcript of the conference. And so I think they may have dragged in a credible scientist and just bastardized what this person said into a completely silly argument. Or this may be somebody who's a complete wackaloon who got a book published. I don't know which it is. One or the other of those is the case. And then they continue. There's possibly both, yes. A whole bunch of conflation of unrelated stuff. Just a rundown of logical fallacy after logical fallacy. And this type of thing is exactly what trolls in comments will do. They'll paste a huge screed of stuff that would actually, to refute all these things point by point, would take you quite a bit of time. And if you do that, then they have won. Because now anybody who's reading this comment is not going to go into the nitty-gritty of the discussion. They're simply going to see that there is a debate.
Right.
And that's all the anti-vaccine people have to do, is sow a seed of doubt. Oh, there's a debate on this between two people who seem to know what they're talking about. Never mind that one of them is completely off the tracks and the other one is actually talking intelligently. You can't tell which is which if you don't know all the background. So my advice to Gary is don't engage any of this. This is something, and I'm absolutely certain that the person who posted this there goes and copy-pastes this exact same screed on any vaccine story they see.
Yep. Yeah. Don't bother, Gary.
Don't get into this, and please stop reading the comments on the news sites that are so stupid that they haven't figured out they need to disable that feature. It's broken. This is why we can't have nice things.
By the way, he cites 35,000 girls have reported adverse side effects from Gardasil.
Oh dear.
Yeah. Well, that's the system working, right?
Yes, that's what the vaccine adverse event reporting system is supposed to do, and that system is horrifically abused by the anti-vax people.
Yep. Look at all these side effects. And then if you didn't have it, they would say nobody monitors.
Nobody monitors it, exactly.
Yeah. Right, thank you, Alan. I'm sorry to make you do that, but
that's okay.
Kathy, can you take the next one?
Sure. James writes, Kia ora, folks. One of the topics that comes up routinely is on trying to place the level of conversation at a level that is not too high and not too low. As a software developer who is routinely talking to non-technical clients, I'll offer a suggestion using a technique I use every day. Try to minimize the number of abbreviations or acronyms used during conversations.
Oh, dear.
The theory I have on this is that abbreviations and acronyms are really useful to increase communication density within groups that know the domain. For people outside of such domains, it can be very difficult to get an understanding when all you see is a wall of acronyms and abbreviations. Restricting such terms to only the most common, like DNA, RNA, PCR, etc., and expanding the others can dramatically help with understanding. This helps improve understandability without really dumbing it down, but rather reducing the data density. Thanks for the awesome podcast, James. And, of course, saying Kia ora, we know he's from New Zealand.
And should we take this as a vote against using FU?
Do we use too many acronyms? I mean, we explained STING, right? CGAMP, CGAS.
Yes.
We tried, but this is what Dickson was doing a couple weeks ago because I think he'd already read this email. Just for example, James gives things from an IT perspective, and he uses UAT, PKI, TLS, PI.
I got it.
We don't know what any of those mean. So if he just spells them out and tells us what the words are, which he does. it's useful.
Yeah,
So
we figure everyone knows what they are, but we're
Yeah.
just in the field. That's the problem.
I'm reminded of a humorous segue that the Car Talk guys had one day. They decided to put aside their normal conversation. It says, now we're going to give you a lesson on writing. So one of the guys says, avoid cliches like the plague.
Yeah, wonderful.
They both laughed themselves silly and carried
Wonderful.
on with their conversation.
Great.
But it's hard to do it when that's part of your normal pattern of speech.
Yep. The next one is from Stephen. He writes, Dear TWiVologists, I wanted to respond to Ralph's letter in TWiV 348 about the possibility of viruses and other microbes escaping in the event of severe damage to a building. I feel like you responded well, but I had an additional, more general take. I don't know what Ralph's thinking is, but the general public tends to think of viruses as nigh on invulnerable. They aren't. Take flu, for example. It's the obvious choice, since it's the center of the gain-of-function controversy, although it's not the hardiest of viruses. As best as I could find from Google, correct me if I'm wrong, under good conditions on good surfaces, which building collapse and terrorist action are not, flu is transmissible for a matter of hours. Fomite transmission can be further reduced or eliminated if the first responders are wearing hazmat suits, which they should anyway. Aerosol transmission is even more limited. I don't know that there are any studies of virus aerosolized by falling buildings, but it sounds like most of the virus particles settle out of the air within minutes after sneezing. Falling building, while it might aerosolize some virus, would also contain that virus as well, it seems to me. The trouble is that society at large has a tendency to think that once a virus is in the air, it's going to float around until someone gets sick off it, which is patently not true. Nonetheless, for instance, one of the characters in Stephen King's The Stand managed to catch the regular flu months after most of the human population had died off, and long after the character had been anywhere near anyone had a case of flu to transmit. Seems to me that this kind of misunderstanding explains why people are so afraid of microbes getting out. Well, he must have got it from a chicken, right?
Right.
And that's why they're afraid of Stephen King also.
I ended up talking about this with another TWiV listener, and we wondered if there have been any studies of lab leaks caused by damage to buildings. Undoubtedly, there have been microbiology buildings damaged by earthquakes and hurricanes and probably other natural disasters. Now, hopefully, all labs are up to code and not liable to crack open, but unfortunately, that won't be the case for some older buildings. As an extreme scenario, I could imagine that an earthquake could knock out of alignment filters protecting airflow out of a lab working with live anthrax. In such a case, you could create an incident like the one in Sverdlovsk, where a Soviet bioweapons program forgot to replace a filter and released large quantities of anthrax. Okay, my hypothetical wouldn't be that much like the Sverdlovsk incident. Volume of anthrax would be significantly lower, and a natural disaster would stop work, but it seems like enough anthrax might get out to cause some problem. So has anyone studied what happens after such incidents? I don't expect anything created in the lab got far, but it would be interesting to know if anything at all has ever been detected or if anyone studied how far microbes manage to get. Are there published studies? The person I talked to suggested the risk management specialists might know more but publish less, so it might be worth talking with one if you could get one on the podcast. They don't seem to want to come on our podcast.
They don't like the odds.
I'm not aware of any studies of disasters of these sort where they look at microbes. Do you, Alan?
No, and it's an exceedingly rare event for any type of lab.
Right.
I mean, and also for any type of building, the types of damage that the anti-gain of function people are hypothesizing is pretty much a 9/11 style sudden destruction of a skyscraper, and it really doesn't happen.
In 9/11, it certainly didn't, but I think the danger is more from asbestos inhalation than it is from viruses.
Exactly. I mean, there was a lot of danger associated with that particular building collapse, but it wasn't because of anything infectious in it, even if there'd been an infectious disease lab.
That's right.
And just in my own anecdotal experience, there aren't that many infectious disease labs in high-rise buildings. Columbia is very much an exception. Usually you go and it's a campus and there's a 2 or 3 story building, and that's where the science center is.
I had a lab at Rockefeller that overlooked the East River, and I also overlooked the island just directly opposite, and I'm blocking on the name
Roosevelt
of it.
Island.
Roosevelt Island. There was a tuberculosis treatment center on that island for many, 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 many years, and TB sticks around.
Yep.
And if you don't sanitize properly and that building falls into disrepair and then they decide to repurpose it or to raise it and to put up another building, that's a possible scenario that might fit into this one, but I think otherwise. And you picked anthrax, too, because that's a spore former.
Right.
Not that tuberculosis is, but it's pretty hardy, and it survives under bubblegum deposits in the subway. So you should think
That's
about
a charming
that, too.
thought.
You know those little black spots that you see
Yes.
on
I know exactly
all
what those are.
chewing gum
Yes.
that people put there, and God knows what's underneath it. I mean, I always thought about that when of course, they're protected because they don't actually come out again. But
Because they're covered with soot.
But now they're repairing the subway. By the way, Alan, they're putting in a brand new subway right here at Columbia Presbyterian Hospital. So
Really?
all of that stuff has been excavated and sent out. I don't know what happens to it afterwards, but you have to wonder.
For the deep station, the 19?
Yeah,
The
109
109,
and the A train.
That's just a surface renovation. They're not doing
but
anything
still,
to the tracks.
but they're beautifying it. They're making it habitable somewhere.
So they're ripping out the tile, or
Yeah, and they're putting in brand new tile. Yeah.
okay. They're not going to air condition it though,
Don't know.
because that's the one improvement that it actually needs.
I don't know of any stations that are air conditioned.
No.
I
Man,
don't
it
think
is
any of them
brutal
are.
down there in the summer
no,
too.
no kidding.
Dickson, did you know there was a smallpox hospital on Roosevelt Island?
A smallpox hospital.
Yeah.
I didn't, but I'm impressed for the fact that that's where they put all of those contagions.
There was another one on what's the island right off Staten Island?
Governors?
Governors.
No, south of Staten Island. There's another one that was another quarantine
Not
facility.
Ellis.
Not Ellis.
Ellis.
No.
No, I've sailed past it now. Anyway, there's another island. It's off the.
It's off Staten Island?
It's off the southeast coast of Staten Island, and it was various sorts of undesirable things, including at one point a quarantine facility.
Oh, I see it here. Let's see what the name of it is. It's very small.
He's got his map.
Yeah, it doesn't have a name. Hoffman Island or
Hoffman. I think it's Swinburne.
Swinburne Island.
Swinburne was
Island
the one
of
that,
no return.
Yes, it was some kind of facility like that.
Lord.
But the scenario that he says that destruction of the building would destroy the pathogen, Alan, you mentioned that last time. You talked about that, how if a building is really destroyed, the viruses, microbes are not likely to survive that either.
No.
Right.
But if any preparedness people are listening, please let us know if there have been any studies of such building disasters.
Data would be good.
Yeah.
Yeah, well, you guys were talking about islands and subways and things. I was thinking about
Kathy was getting some work done.
about
Did 3 transfections, 4.
hurricane or tropical storm Allison that damaged research mice in Houston,
Right.
and Hurricane Sandy and the mice at NYU. So the fact that it can affect lab animals,
For sure.
it could
And
definitely.
Katrina damaged hospitals and labs in New Orleans, of course.
Yeah,
Right.
and what about Fukushima? I wonder, that huge earthquake and tsunami that followed, I wonder what the consequences, long-term consequences.
Maybe the radiation killed all the bad bugs.
But these are all these are not in high containment facilities. They're not in BSL-4.
No.
So,
Right.
so there is a 4 in Galveston, which has got a lot of hurricanes and floodings. And as far as I understand, it probably wasn't a great idea to put it there, but it's been intact, right?
Yeah.
Right.
Right, but there's enough nasty BSL-2 things that,
I have to beg out.
Bye. Do you have a pick?
I do have a pick.
We were going to let you read the last email, but that's fine.
Okay.
What's your pick?
My pick is a new movie that's come out. It's called the PhD movie. It's based on the PhD comics. And I know that was a popular subject, and now they've made it into a full-length movie that's about to be released into the local neighborhood theaters. So I don't know what it's like, but I'm probably going to go see it just to see if being a graduate student today was anything like when I was in school, but I probably think it's not. But
It's not
nonetheless,
Dickson.
the anxieties and successes and failures of muddling through 4.5 to 5 years worth of indentured slavery is worth revisiting. And I think people deserve to know what it's like to be a student dedicated to some esoteric subject, which, eventually 10 other people will understand what you're doing. And you have such passion for that subject that nothing can deter you, including personal income, personal life, etc., comfort, the whole thing. We put everything aside to pursue knowledge for its own sake in some cases.
They're making a sequel already.
They're making a sequel already.
PhD movie 2. They raised $163,000 on Kickstarter.
Well, they're going to have to follow up on this one, and it goes for the postdoc after that. Then that's just, then young faculty member.
Cool.
Yeah,
Thank
so
you,
that
Dickson.
was my pick.
Goodbye.
I'm sorry, I have to leave now.
Have a nice weekend.
And everybody else, have a great extended holiday, and see you next
too.
Friday.
Bye-bye, Dickson. Don't let the door bang you as
I know what you're saying.
out.
Okay. I take such abuse over here.
Well,
And
yes.
we're just going to make one more comment on the risk analysis people. I have the feeling that there was somebody who wrote to us from California who has written several times about various risk analysis things that he's had to do in his job, and he might be interested or willing to come on and talk if we ever want to have somebody like that. He could write back and tell you who he is.
Then there's the Sandman Institute, which is around here, and they have written before, and we've talked about having them. They do risk assessment type things. So, yes, we should have people talking about risk assessment. That would be good. All right. Goodbye, Dickson.
Goodbye.
Let's do some more picks of the week since we've started. Alan, what do you have for us?
Okay. Let me just scroll down. So I have picked, this is really a photography project and not a science project, but this guy's creativity is just amazing. It's called Miniature Calendar. And this fellow poses miniature figures in various settings, like they're doing things. So, for example, the one he has, he's based in Japan, so he's already posted the one for Saturday, the 5th. And this one's called Southern Island, and he's got a plate of, looks like meatballs with gravy and some maybe thyme or something, and a fake palm tree that forms an island. And then he's got a woman in a bathing suit reclining on the top of one of these things, and a man standing down on the puddle of gravy like he's standing on the beach.
It's great.
And they're all like this. And what's amazing to me is that this guy does one of these every single day, and he's been doing it for years.
Wow.
And I just came across it recently, but you can scroll back through, and it goes, I guess, to 2011. He started doing this, and he just keeps going.
Wow. These are neat.
Yeah, they're very cool.
Yes.
Take a look at them. Yeah.
Yeah, he does a really good job staging them and photographing them, and it's just all these tiny little figures doing these funny things with ordinary day-to-day items.
Yeah, the camel on the sponge is good.
Yes.
So
Very cool.
This thing of doing something every day is pretty interesting. A number of artists have done that, right?
Yes.
So Jonathan Coulton wrote a song a day for a year. And recently we had something else. I don't know if it was a pick or something. Someone did something once a day, which went on even longer. I can't remember what it was.
Yeah.
Yeah.
Well,
maybe I'll do a podcast today.
Huh.
That's
You'll have to wait till after you're emeritus. to do that.
true. If I ever am emeritus, right? Kathy, what do you have for us?
I picked something that I found out about after the ASV meeting, or during the ASV meeting, kind of indirectly, but afterward, more details. So you might remember that there were a couple of PLOS pathogens staff people there, and I asked them what they do in their spare time. And Lily is quite the runner, and Laura said something about, I do some large art that involves welding and things. So afterward, I wrote to her and said, tell me more about this. And so she sent me this link, which is to Dr. Brainlove 2.0, and there's a couple of Vimeos. The most recent one is the one that I've labeled here, number 2, with narration explaining it. And it's a group of neuroscientists in the San Francisco Bay Area. I think there's at least a dozen of them that are neuroscientists, and then there's others, scientists, and they've made this huge brain that's been to South by Southwest and Burning Man, and this year they figured out a way to make it modular so it's easier to put together. It's got a dance platform in it, but but it's just really cool because it's modeled after a real person's brain, a woman who's in the group, and they have all these lights. So it's not just a cool thing that they've built in a group thing for them to get together and do this, but it's anatomically correct and they can show things and do things with it. So it's really worth checking out. And then I watched one Vimeo and I wrote back to her, and then she said something about, I don't know if you could see it, but there's a 6-foot bacteriophage on the top. And that's because the group is called the phage. They're all scientists, either practicing or former or something like that. And so I hadn't seen it, but she sent me the picture, which I put here in the show notes. And in one of the Vimeos, just today, I rewatched it, and I knew where to look, so I saw it. And that's at 352 in the second one. But
it's clearly out of scale.
Yeah, compared to a human brain.
Which is a funny thing to say to when you're talking about a human brain that's built onto the back of an old school bus chassis, but
right.
You know what I mean.
Yeah. Anyway, so I think it's just really cool. It's a way to tell people about science, particularly at South by Southwest and Burning Man and the Bay Area Science Festival, things like that. And it just really seemed like a cool group activity and worth telling you all about.
I wish they hadn't put a phage on the brain, though. I understand that they're called the phages, but, has Laura actually helped make these things?
Yeah, she's in one of them. If you
cool.
watch, I think the one that I've labeled number 2, no, I don't know, number 1, I think she's in that
cool.
one for sure. Yep, so
Yeah.
watch for her.
I remember her from ASV. Sure, did a lot of selfies with her and her friend Laura and
Lily.
Mm-hmm. Nice people. Thank you. My pick is a browser extension called Ghostery. Now, there's a big brouhaha in the IT world about advertisements on web pages, both desktop and mobile. They are really messing up the experience. Basically, when you go to a website on a browser, all these scripts load, and they decide which ad you're going to get. And it can really mess up your browsing experience. It can add megabytes to downloads. It can persist for many sites afterwards. It's really a problem. So a number of solutions have been developed to deal with this, and this is one of them. It's an extension you install, and then when you go to a website, it shows you all of the JavaScripts that are loading. They're basically ad trackers and loaders and so forth. And you can decide which ones to enable and which ones to disable. I find it interesting because it has really sped up the browsing that I do. You can use it for Chrome, Firefox, Safari. You can use it on mobile as well. And some pages you go to, you got 20, 30 different scripts loading. And sometimes they take a few minutes.
Wow.
So I listen to a lot of tech podcasts, and this is obviously going to change the way ad works because the people who do the advertising distribution just don't realize they're screwing up people's browsing experience. They're going to have to figure out another way to pay for content that doesn't involve being obnoxious. In the old days, it was pop-up ads, so browsers incorporated pop-up blockers. And now we have these JavaScript ads. So I've been using Ghostery. It really helps because here at work, I just want to get work done. I don't want to have to wait for a page to load. And you can pick what to load and what not. I respect that some people's pages need ads to work, but they certainly don't need 18 different scripts.
So you do it on the fly for each time a page is loading?
So when you go to a site, it will show you all the scripts that are loaded there. And then there's a preferences that you can say, on this site, I only want these to load or none.
Okay.
And then when you go back to that site, it will only load the ones that you want. So it remembers them, yeah. Mm-hmm. So I find it useful because you can really cut down on the time. And if you're interested, if you're concerned about privacy, I'm not too concerned about the privacy that'll deal with that. I just am concerned about the time. Sometimes my browser is just crashing because so many scripts are loading. And you'd be surprised at what sites are loading tons and tons of different scripts. So anyway, ghostery.com. Check that out.
Have you used AdBlock Plus at all?
My son's installed AdBlock on Chrome, yeah.
Okay. I was wondering about the comparison because I've been using AdBlock for several years, and it makes a huge difference in page load times and just general readability of the web.
So this one will also block non-ad scripts that are doing tracking and all that sort of thing. I don't mind having Google Analytics, right? Because I understand that it's important to get data, but there are lots of other trackers that load that. Why do you have to do this? So it puts a little thing in your browser bar, and you can click on it and see what's on the page. And so right now I'm on the show notes for TWiV 353, and there are no trackers, which is very nice because we didn't put any on. So check that out. We have a couple of listener picks. One is from Ramon, who sends a link to a really nice New York Times article. Children die because people are wrongly afraid of vaccines. And this is the sort of thing we need more and more of. This is an opinion, of course, but the more this gets done, the better. And we, of course, heard about this business just today. And while we were recording TWiV, I got a text message from Chris Kandian, our friend at ASM. He wanted to talk to me, and I said, I can't. I'm TWiVing. So he said, okay, call me later, but here's my pick. And he sent a link to ASM Live at ICAC, which is coming up in a couple of weeks in San Diego. It's on the 17th of September. ASM will be doing live interviews again with Michael Schmidt, so I'll put a link to that in the show notes. So thank you, Chris, for that. And that is it for TWiV 353, which you'll find on iTunes and TWiV.tv. And please do send us your questions and comments to TWiV at TWiV.tv. I want to thank Dickson Despommier. He's gone, but he is at verticalfarm.com. Thank you, Dickson. Always a pleasure. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Alan Dove is at turbidplaque.com and also on Twitter. Thank you, Alan.
Always a pleasure.
And I am Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.